I'm going to go through the phases of the cell cycle again, mainly though mitosis. So we've already gone through G1 is where most cells are spending their time. The S phase for synthesis of the DNA. G2 is where there's more growth. And then there's prophase, metaphase, anaphase, teleophase, and then cytokinesis. So diploid cells divide by mitosis. Each new cell receives one copy of every chromosome that was present in the original cell. It produces two new cells that are both genetically identical to the original cell. In other words, they are clones. Okay? So we have a diploid cell down here. This is a duplication of all the chromosomes during the S phase of the interphase. So we've got double of each. And then we've got mitosis, where these two are pulled apart and each daughter cell gets complete complement of DNA. This is the mitotic division of an animal cell. The one on top is actually photographs of it. On the bottom is just showing everything. So we start with the end of G2 of interphase. So the cell's been growing and it's ready to start doing mitosis. As you can see, we have centrosomes and these centriole pairs, and they will split apart. All the chromatin inside. The outside is the nuclear envelope, which is going to break down. Plasma membrane. There's these nucleolus in here, you can't really see them. And then on prophase, this is the beginning of mitosis. The early mitotic spindle starts to form. These are along here. And they're attached to each centriole, here and here, and they form an aster. Now the centromere, of course, is the center of the chromosomes that are duplicated. And then there's, of course, the chromosomes with two sister chromatids. Okay. Now, in prometaphase, a number of things happen. The nuclear envelope has started to break down. Of course, the centrioles have gone to either end. We see, one second. We see these kinetochore tubules, these things, they're stretching out, and they're attaching to the kinetochore of the duplicated chromosomes. And then there's also these non-kinetochore microtubules. But each of these chromosomes is attached to two microtubules, one from one side and one from the other. We start with metaphase, and what first happens is what's called a metaphase plate. It's an invisible plate right through the center of the cell. All the chromosomes have lined up along the metaphase plate. We've got a centrosome and one spindle pole at each hemisphere. And then we've all got the spindle fibers. So now, during anaphase, the daughter cells split apart, or taken apart. So each, daughter chromosomes. So each chromosome is going to be pulled to that end, to that end, that end, that end, all that way. Okay? Anaphase is the longest actual phase of mitosis. And then finally, during teleophase and cytokinesis, we've got each chromosome, each cell has complete chromosomes. there's this cleavage furrow that we see right there, and then nucleolus and the nuclear membrane is forming. And then you're going to end up with two daughter cells. Each of these following slides is going to go through each of the phases. So this is the end of G2 phase. There's a nuclear envelope. The nucleus contains one or more nucleoli, singular nucleolus. Two centrosomes are formed. Chromosomes duplicated. And then we'll go on to the next. Prophase, again, you can read this on your own. But we end up with the centrosomes. We've got chromosomes that are consisted of two sister chromatids, and the nuclear envelope is starting to break down. Now we're at metaphase. The metaphase plate has formed, of course, and you saw this all before, so you can see what's happened. This is the longest phase. And then, I want to go over the mitotic spindle. So we've got these microtubules and the chromosomes that attach to the chromosomes. Overlapping non-kinetochores. Okay, so the kinetochore microtubules look like this, and they attach, and they're pulled to each centrosome. Now in anaphase, finally, the chromatids are being pulled apart so that each cell will have a complete chromosome or genome. Okay? And then, of course, telophase, this is where they split into two daughter cells. They're identical. Okay. You can see this, let's say this cleavage furrow in an animal cell. This is a plant cell. It's different, looks different. The wall of a parent cell. So the same thing happens with a plant cell. And you end up with two new daughter cells.